Good evening, Southport. Fiat Lux, Let There Be Light. As you can see, this is part 4. You don't need to know about the other 3 parts. This is a trilogy, and therefore this is part 4 of the trilogy, which I decided to write after the first 3 parts. This is Colour in the Cosmos. I said a little bit about colour in talks I've given in past years, but I thought colour was so important it deserved a talk all of itself. So this is about colour and the universe in colour, but I guess you could argue that this talk is more about colour and a little bit of cosmos. So we'll see how it goes when we split it. I'm going to give a short introduction, and I'm going to be spending the first maybe half of the talk talking about how we perceive colour, what colour is, how we perceive it, how we quantify it, before moving on to, so why do we have stars that have particular colours, and why do we have nebulae that have particular colours, and why are they different? If they're all made of the same stuff, if the gas out there is the same as the gas in the stars, why are the colour of nebulae not the same as the colour of stars? I don't think I'll have too much time to talk about spectroscopy. That will be covered in some of the slides, which you're free to pick up at the end if you so wish, but I won't be saying too much about spectroscopy in this particular talk. And then I'll finish by looking at beyond colour, and then summarising right at the end. So we can start with the obvious question, and that is colour real? Or is it just a pigment of your imagination? And yes, when I tried putting that in, PowerPoint tried to change pigment back to figment again. It was trying to be far too clever. So why do we have colour vision in the first place? Well, apart from the fact rainbows would be really boring if we didn't have colour vision. Just imagine that particular image there. If we didn't have colour vision, it just would not be the same. Many people believe that we have colour vision in order to be able to tell, for instance, when fruit is ripe. So here, for instance, there are 3 fruiting bodies there. Anybody care to guess which, if any of them, are ripe? Can you tell if you haven't got colour vision? No, pretty difficult. Whereas if you have got colour vision, it is pretty obvious which are the ripe ones and which are the ones that are unripe. Perhaps I should have asked right at the start of the talk, is anybody here colourblind? No, okay. So I don't have to apologise to any individual member of the audience for not getting the most out of this talk. Okay, so you might argue that we have colour vision in order to, for instance, pick the right food. I don't think that's right. I think we have colour vision so that we can make sense of maps of the underground. Otherwise they're really difficult to interpret. As soon as you add colour, then you can say, yeah, it's starting to make sense now. I can see where I need to go. So maybe that's why we've developed colour vision. So how do we perceive colour? I'm sure a number of you will already know this, but we'll go through the basics anyway. So in the human eye, light, of course, is focused by the cornea and the lens onto the retina. And the retina, which covers most of the back of the eye, obviously it has a huge field of view in terms of angles. Most of the back of the eye is peppered with photoreceptors. Some of them are colour sensitive, some of them are not. So the so-called cones and rods are the different types of receptors. The cones are the colour receptors, and we can think of them as having red and green and blue colour receptors on the back of the eye. We'll talk about rods in just a moment. But in terms of colour, it's those that matter. And those colour receptors basically pick up the light, convert them to a chemical signal or an electrical signal, which are then fed down the optic nerve to the brain. So that's in principle how we perceive, but of course there's far more to it than just the photochemistry of what's happening with photoreceptors. If we think of these photoreceptors and say, well, isn't that exactly how a camera works, a colour camera? In the days of film it was a little different, but now we're working in a digital age. You could say that if we look at what's going on in the chip of a colour camera, if we look in detail at what's going on, we see that there are pixels in the chip. None of those pixels are themselves colour sensitive. All CCDs, all charge coupled devices, are basically colour insensitive, and we get the colour sensitivity simply by putting filters on top of each pixel. And usually the pattern is this one. It doesn't have to be this, but this is a very common pattern called a Bayer grid, in which you have blue filters, green filters, and red filters in a particular pattern, and the computer knows which is which, so it knows this pixel is picking up light that came through this red filter, this pixel is picking up light that came through this green filter, etc. And then the computer can reconstruct a color image based on that. If we ask ourselves, well, what light is actually transmitted by each of these red, green, blue filters? I've taken a camera that I actually owned a few years back. It's a Nikon camera, it doesn't really matter which one. And we can see that the light coming through the blue filter lets through, very roughly speaking, the blue end of the spectrum. 1/3 of the spectrum, the blue end, is let through by the blue filter. The green filter lets through, very roughly, the middle third of the spectrum. And the red filter lets through, well, a little more than just the red third of the spectrum. But very roughly, you can see that they are, broadly speaking, letting through 1/3 of the full spectrum of light into each of the red and green and blue filters. And you might say, well, human vision surely looks pretty much the same. If we were to map out how sensitive our eyes are to the different wavelengths of light from the blue end of the spectrum to the red end of the spectrum, here it's written in terms of the actual wavelengths of light, 400 nanometers on the left, 700 nanometers on the right. For most people, that's the range of human vision. There's obviously a little bit of variation from person to person, but that's a fairly good average. It's tempting to think that human vision surely looks the same if we were to draw the same curves for the human eye. Turns out not. They look rather different. It's surprising just how close red and green are to each other. Not only that, notice that blue looks perhaps similar. The blue is the bottom third of the spectrum. But look, red and green, for instance, they both have some sensitivity at the extreme end, and red indeed goes all the way to the other end of the spectrum. So you could argue the red receptor, which you might want to call the red receptor, is actually sensitive to all wavelengths of light. And where is it most sensitive? Well, it's actually not most sensitive in the red. It's actually most sensitive in the yellowish, greenish. That's a bit of a surprise. Green, not surprisingly, is most sensitive in the green, and blue is most sensitive in the blue. So although it's tempting to say, well, these are analogous to the red, green, blue, or the RGB or the BGR filters in a camera, biologists don't refer to these three color receptors as the red, green, blue color receptors. That's what we might tend to think about, especially if we're used to using cameras. But biologists don't refer to them as red, green, and blue. The biologists refer to them as the long wavelength, the short wavelength, and the mid wavelength, because red, green, and blue is a little bit misleading. So despite that, I'm still going to call them the red, green, blue, because I'm so used to doing that and so used to thinking about red, green, blue receptors in the eye. Why are they so close? Well, that's probably an evolutionary trait. One of the reasons they're quite close is that for quite a lot of wavelengths, there is a big difference between the red and the green. Even though they seem to be quite similar in that they overlap considerably, there are plenty of differences in the intensity of the response between red and green, and that's perhaps what gives us our particular color sensitivity. But whilst we're here, it's worth pointing out that that's where the color receptors of the eye sit. But remember, there is another receptor in the eye, the rods, that are not color sensitive. They actually sit there. Let's just get rid of the RGB so we can see them a little more clearly. The rods in our eye are sensitive at night. They're not particularly sensitive. The cones are not particularly sensitive at night, which means the rods come into play at night. So the rods are sensitive to low light levels. To see color, we need plenty of light. The cones do not operate particularly well, if at all, at night. And it's only the rods which are not color sensitive, because we don't have three sets of rods. We don't have RGB rods. We just have one set of rods. And the color sensitivity, you can see that they are mainly sensitive to the blue and maybe to the green part of the spectrum. And that is why we use red lights at night. Because if we were to use a red light, if we want to be able to see something clear enough, we want some bright light. If we use red end of the spectrum, the rods don't even know that that red light is there, because the sensitivity of the rods has basically died by the time we get to the yellow part of the spectrum. So if we use red lights at night, even a relatively bright red light, we will still preserve our night vision, because the rods will not be aware of that and will not change their sensitivity. They will not reduce their sensitivity on seeing a very bright light because they will basically not see red light at all. So that is why astronomers use red light. If the rod sensitivity went all the way to the right, we would not be able to use any light at all at night without affecting our night vision. So it's just as well the rods are over to the left-hand part of the spectrum there. We have 3 photoreceptors, red, green, and blue. Some animals have more. Just because we can live with 3 and we think 3 is all you need, any color can be made from a mixture of red and green and blue. Why on earth would you need any more than that? Some animals have got a considerably more complex set of eyes. There's 700, which is where our sight normally finishes. So this animal can also see in the infrared. And remember, 400 is the blue end of the spectrum for us. So this animal has actually got a few receptors that actually go all the way into the ultraviolet. Would anybody care to guess which animal this is? Nope, not a cat, not an owl. Interestingly, that you picked insects, because a lot of people said either an insect or a really weird animal. And the weird animal I can think of is an octopus, because they're always weird. They've got loads of brains and loads of hearts and loads of legs. In this particular case, it's actually a mantis shrimp. Why would a shrimp need to have more than a dozen photoreceptors, types of photoreceptors? It's got so I presume it's to see its food. Quite why it needs that many. Perhaps its food shows up in the ultraviolet in ways that we can't understand, but quite why it needs so many. Why doesn't it just have 2 or 3 photoreceptors of which one is in the ultraviolet? I don't know. Maybe there's a good evolutionary reason why mantis shrimp have evolved with that many photoreceptors. The point is there's nothing particularly special about 3. We tend to think of 3 as important because colors can be made by combinations of red, green, and blue primary colors. And we tend to think of that as being universal, but it is by no means universal. So why have we got 3 at all? How did we end up with 3 photoreceptors? Well, we haven't always had 3 photoreceptors, or at least we haven't had 3 working sets of photoreceptors. It goes all the way back to where we came from millions of years ago. When dinosaurs ruled the Earth, for instance, we were a small mammal about yo big, small furry creature, and we were nocturnal. We only came out at night because if we came out during the day, we were either eaten or stamped on by a dinosaur. So we stayed indoors, we stayed underground, came out at night. We needed night vision, so it was rather important to have rods in the eyes that were sensitive at low light, color not so important. So at the time, we didn't have 3 receptors. But over a period of, I don't know, 100 million years or so, evolution has taken our color vision back again because we started to become diurnal. Fairly soon after the dinosaurs were wiped out, we suddenly realized, hey, it's safe to go out during the day now. And so we started to go out during the day, and then color vision became more important as we started gathering food during the day rather than during the night. So genetic mutation has basically given us our 3 color vision back again from what we had many millions of years ago. I'm not claiming that's the pinnacle of evolution. Of course, Homo sapien isn't the end of the evolutionary tree. Who knows where we will evolve from this point onwards? I mean, it's not clear where we're going, but yeah. And we know as astronomers that if we look at what light comes through the Earth's atmosphere, again as a function of wavelength, thinking of the whole of the electromagnetic spectrum, our atmosphere happens to be transparent in the visible part of the spectrum, so the sun produces most of its light in the visible part of the spectrum, and that light can reach the surface of the Earth, which of course is why we've evolved to have sensitivity over that particular range of 400 to 700 nanometers, because a lot of that light will actually reach the surface of the Earth. Lots of shorter wavelengths will be blocked, lots of longer wavelengths will be blocked. There's lots of spikes in there because different molecules in the Earth's atmosphere will absorb or block particular wavelengths of radiation. There happens to be another window when we get as far as radio waves, but we're not going to be talking about that this evening. So if we want to observe the universe in the short or long wavelengths, we are happy to deal with telescopes on the ground for visible, but if we want to see what's going on in the UV and in the infrared, we need to get above the atmosphere. So we've sent up lots of satellites. Ultraviolet and X-ray satellites have gone up, and infrared satellites like Herschel, Spitzer, and James Webb Space Telescope are looking in the infrared. Of course, that doesn't stop us, if we so wish, putting a visible telescope up above the atmosphere. That's not because the light doesn't reach the ground level. 90-something, maybe 99% of light does reach ground level, so we don't need to put a telescope in orbit in order to see the universe in the visible part of the spectrum, which is just as well, so that we as amateurs can carry out astronomy. But of course, the Hubble Space Telescope is mainly above most of the atmosphere. It's very thin at the altitude that it's orbiting. It's not above all of the atmosphere, but it's above most of it. And so, of course, it's the turbulence of the Earth's atmosphere that the Hubble Space Telescope is getting above. It's not the absorption of the atmosphere, it's the turbulence giving us a less than clear view. So if we return to this idea of the human eye, I said that it's based on red, green, blue receptors. Not the correct label, but that will do for the time being. There are a whole load of photoreceptors. The color ones are the cones, the rods are the low light photoreceptors. And it is, not surprisingly, it's far more complex than I've indicated there on that little cartoon of simply having red and green and blue photoreceptors lined up on the back of the eye. What's actually going on there is far, far more complex. We've got lots of cells that are photoreceptive, in other words, they are sensitive to various wavelengths of light, but there's also a whole load of other cells in there as well. So the photoreceptors are this layer at the back, but sitting on top of them, there's a whole load of other cells called neurons. Now, perhaps you think of neurons as being, well, isn't that what the brain is made of? Well, yes, neurons or neuron cells are in the brain, but there's a whole shed load of neurons in your eyes, and they are doing an awful lot of image processing. So this idea that you see light because light hits the back of your eye, the back of your eye picks up the light and basically says, well, there's this much red, this much green, this much blue, send the signals to the brain and the brain figures out what you're looking at. No, that's not actually how it works. The neurons, these cells in the back of your eye, are actually doing a lot of image processing long before those signals actually get to the brain. So in a sense, your eyes, neurologically speaking, your eyes are part of your brain. They have a whole load of neuron cells that are doing some processing, which is then fed to the rest of your brain to do the last bit of image processing. And we can see what that's like. This is a sort of schematic just to sort of try and solidify that particular point. If we ask what's going on in a camera picks up light on the chip, having gone through each of the colored filters, red, green, blue filters. That hits the pixel. That records how much light has arrived. And so the camera is effectively recording how much red, how much green, how much blue light has hit the camera when you've taken your picture. How is that converted to an image? Well, it's essentially a one-to-one circuitry in the camera is basically taking the red hitting the chip and it's using that to generate the image which ultimately gets saved on your SD card. Similarly for the green, similarly for the blue. Yes, it might adjust the levels a bit. It might do a little bit of a color balance because maybe there's a bit too much red. Maybe this camera chip is quite sensitive in the green and it needs to be backed off slightly. But roughly speaking, the signals hitting the chip, the red, green, blue signals, are taken to make the red, green, blue elements of the color image that you ultimately see. The brain doesn't work that way. If we say what happens with the brain, again, we're picking up red, green, blue signals, but those signals are then fed through a neural network because your eye is a collection of neurons, so it is a neural network. The brain is a very sophisticated neural network. The eye is a slightly simpler neural network, but this is not a one-to-one correspondence between how much red, how much green, how much blue is detected versus what the brain is actually told about what's actually going on. So these lines and so-called nodes in this network are supposed to represent the fact that, for instance, it might be that the sum of the red and the blue is important, or maybe it's the difference between the red and the green that's important, or maybe it's how much has blue changed since we last looked at blue half a second ago. These things are being processed by these neurons, and then the output, which can be very complicated, is the output sent to the optic nerve, which is then sent to the brain. So the brain does not receive red, green, blue information. The brain receives information such as what has changed, what's different, what is brighter than what. And we can see that in action, or at least we can see the results in action, if we do a little test, which we're going to do here. If you want to check this for yourself, the web page is given in the bottom left corner there. We're looking at a couple of sort of Rubik's Cubes type things. And I think you can see that there's a whole load of tiles. What is it? It's 5 by 5 tiles. And there are different faces, but I'm only interested in what's going on the top face of these cubes. So I'd like for you, if you can please, to count in those 25 tiles, the top face of the left-hand cube, how many of them are blue? Anybody disagree with 4? Everybody see 4 blue tiles? Okay, let's do the same on the right-hand side. Again, we've got 25 tiles on the top face of the right-hand cube. How many of them are yellow? So it sounds like a consensus is there are 4 blue tiles on the left, and there are 7 yellow tiles on the right. Yeah? Okay. Are you prepared to put money on that? That has instantly seeded some doubt in your mind. You may be interested to know that the 4 blue tiles on the left and the 7 yellow tiles on the right are actually the same color. All 11 of them are the same color. Even though you think the 4 on the left are blue and the 7 on the right are yellow, they're not. And what I'm now going to do is remove all of the pixels other than those 7 and those 4. And I'm going to take those pixels away one step at a time. That's what you were actually looking at. They're not blue. They're not yellow. Your brain, or your eyes, we have to always say the eyes and the brain, were interpreting these grays as blue, and your eyes were interpreting these grays as yellow. Why? Because the background pixels were confusing the issue. What your brain was actually told was these 4 are bluer than most of the other pixels I can see in the same field of view. And your brain was told these pixels are yellower than most of the pixels I can see around them. Your eyes were doing the processing and picking up signals here, signals there, looking at differences, contrast, variations, and telling your brain what it thinks your brain needs to know. Your brain is not fed with absolute intensity information. It's not fed with absolute color information. It's fed with information that, by evolution, we think your brain needs to know. It needs to know whether that orange thing over there is a lion in a bush or not. It needs to know whether or not something is coming into your peripheral vision. It is not important to know whether or not that red is absolutely 56% brighter than that green. That's irrelevant. What matters is differences and changes. So that example shows you, if I just do it again, take away all of the other pixels, that's what you are actually looking at. And if you think I'm cheating, you can go to that web page and prove it for yourself. I would never lie to you. And although it's strictly not color, this is another very nice indication of how image processing is being carried out by your eye. So it's just another point about neurons, not specifically about color. I'm interested in the relative brightness of the square labeled A and the square labeled B. Not the letters themselves, the square on which the letters are sitting. Would you agree that one of them is brighter than the other? Okay. let me, in a slightly different way, block out all of the pixels that aren't relevant in this image by simply masking them out. Now look at the grey there. It's exactly the same as this grey. Look at the grey for A. It's exactly the same. Your eye, if you wish, can simply wander around between A and B and convince yourself they are the same. They're not similar, they are identical. They are absolutely the same shade of grey. I've gone over this with a computer program and checked the values of the pixels. They are all exactly the same. So square A and square B are exactly the same shade of grey. Let me take those blanks out again. Okay, what's your brain telling you? What are your eyes and your brain telling you? You absolutely know that they're the same shade of grey, because I've just shown you they're the same shade of grey. But even though you know that's the case, your brain refuses to allow you to see that, because it's trying to tell you what it thinks you need to know. And it's being fooled by the fact that this green object there is casting a shadow, and it's trying to interpret what you're seeing, telling you that although they might be the same shade of grey, I'm trying to tell you that because there's a shadow there, that is actually brighter than that, which is usually what you need to know. This is a sort of optical illusion that's been designed just to highlight how much processing is done by your eyes. So if nothing else, the take-home message from this talk is you cannot believe anything you see. Nothing to do with AI fakery, simply things that are on the screen are not necessarily what you think they are. So how do we quantify color? We find it difficult to perceive. It's very difficult to say that looks different to that. We can always say that looks redder, but different people will have different opinions. How do we actually quantify color? If you ask a physicist how to quantify color, the knee-jerk reaction is, well, you look at the spectrum. Now, for some of us of a particular age, that's a spectrum. But what I'm talking about, of course, is that spectrum where Isaac Newton, back in the late 1600s, was trying to reproduce the album cover of Dark Side of the Moon by sending light through a prism and having a look at the result. So we've known for quite a long time that white light, what we would call white light, comprises many colors, and we can split them with a diffraction grating or a prism or other optical instruments and separate out the colors. And essentially, any optics will do that. Prisms are designed to split light. Diffraction gratings are designed to split light. But almost anything other than a pure reflection of a perfect mirror will reveal the colors of light. And almost any optical instrument will reveal that that light is made of waves. It's almost impossible to get around that particular limitation. So we can think of waves. The wavelength is defined there. The separation of two, you can call them crests and troughs if you want to think of them like water. That's not quite the way it works with electromagnetic waves, but it doesn't really matter. Wavelength is the repeat distance, if you like, from one peak to the next, or one equivalent point in the wave to the next equivalent point. And as we've already intimated, when we're dealing with the visible part of the spectrum, then we're running from approximately 400 nanometers, a little less than half a micron, to 700 nanometers, a little more than half a micron, are the wavelengths of visible light. There's nothing particularly interesting about 400 and 700. That just happens to be the way we measure distance. That's what the numbers happen to be for the light coming from the sun that resulted in our particular sensitivity to wavelengths over that range. But of course, that can't be all there is to colors. In other words, all colors are not all the colors of the rainbow, even though we talk about all the colors of the rainbow. Because, for instance, when we look at the rainbow, where's beige? Where's brown? It's not in there. So the colors of the rainbow are only a certain range of colors, not all colors. So if we want to somehow map out all colors, we can't just put them in a line from red to blue and have a continuous range from red to blue, because that doesn't include other colors. So we have to find a different way of laying out colors that don't simply result in them being in a one-dimensional line. And perhaps you can guess what we need. We need a two-dimensional way of mapping out colors. And a long time ago, back in the 1960s 60s, I think it was decided this is one way of doing it, and it's now become a standard way of mapping out colors in a two-dimensional plane. In this particular case, we've got a two-dimensional color space, as it were, and I am not going to go into what these axes actually are. You can define colors in different ways. You can say, well, let's look at the amount of red and let's look at the difference of blue and green, and we can map it out in different ways. This particular commission, this International Commission on Illumination, decided that of all the ways of doing it, this is not necessarily the best, but at least it's a standard, and everybody can agree that we can map out colors in this particular way. So again, we're not going to worry about that, but you can look up CIE if you want to know more details of how they actually established that this was a good way of quantifying all colors. So now, in principle, all colors should appear in that particular color space. The spectral colors, the colors of the rainbow, how do they relate to this color space? Well, the colors of the rainbow are on that outside arc, running from red on the right all the way through green at the top down to blue at the bottom there. Notice that there are no spectral colors between 700 and 400. There's a line here which goes through purples. There's no purple as such in the rainbow. There's violet at one end and red at the other, but there's no purple in the rainbow. So the rainbow wraps around the top, and there's a shortcut at the bottom which gives us other colors which result from mixing red and blue together. Notice that they're not equally spaced. That's not a requirement of this particular color space. What matters is each color has a unique position in this two-dimensional space. It doesn't really matter that the spectral colors are stretched out a bit. In the case of over here, for instance, 20 nanometers is a big stretch, whereas down here, 20 nanometers is a relatively small distance. Now, one catch is I can already see that there's a difference between what's being displayed on the screen and what I can see on my laptop. And that's because every physical device, every TV, every tablet, every data projector has a different way of displaying colors. If you think about it, how do you actually get these colors on the display? Well, this particular projector is taking a bit of red and green and blue and mixing them together in particular ways to try and generate every color that I want it to generate. But given that every projector and every tablet has a limit to the range of colors that it could possibly display, that means there's no guarantee that when you look at an image on one display, one TV, one monitor, one tablet, you'll necessarily see the image looking the same when you look at that same image, even though it contains exactly the same information on a different device. Moving from a tablet to a desktop computer to a TV, things can look different. So whenever you're displaying colors, for instance, in this particular case, we have a limited gamut of colors. A gamut is simply the range of colors that it is capable to display. And it's resulting from mixing together red and green and blue colors. So if you like, that red, green, and blue position there are the three primary colors that are used to mix all the other colors. But basically, you can only mix those together to generate colors inside that triangle. Using that red and green and blue, nothing you can do with those three colors can possibly give you colors outside the triangle. So manufacturers of particular devices will try and get their red of their projector red, for instance, as close to that corner as possible. They'll try and make the blue as close to the corner as possible. They'll try and make the green as high as possible. And if they can do that, you get a nice range of colors. That's what TV manufacturers are always trying to do, to try and get red, green, and blue as far apart as possible so you can mix all the colors you need. You can probably see on this particular projector that all of those purples down at the bottom look very similar. They are not distinguishable because the projector cannot distinguish this color from that color from that color. Notice that to the left of what I've labeled green is a sort of a cyany color, but the whole range looks cyan. You can't really see differences in different parts of that color space. And that is a limitation of the projector or the screen or the tablet or the TV, whatever it is you happen to be using. And it's different every time I give this talk. The appearance of that slide keeps changing every time I give this talk, because it's usually with a different projector or a different TV every time. Okay, let's have a think about the cosmos. We've looked at perception, we've looked at how we quantify colors. Let's think about what the universe is doing in terms of making colors. If you ask yourself, where do stars fall in that? If you were to map out various stars, if you were to look at stars and calculate their or measure their colors, and then started plotting them on this diagram, where would they be? Would they be scattered everywhere? Would they be mainly on the left, mainly on the right, mainly at the top, or would they be scattered over the whole area? Well, it's perhaps surprising to know that because of the laws of physics, stars can only fall on that line. They can't be any arbitrary color they want to be. Stars have to be somewhere along that curved line. Cool stars are on the right, and hot stars are on the left. So in other words, if you were to line up stars according to their temperature, you would see them either as red or slightly orangey or relatively white or a little bit bluey white. You don't get green stars. There's no such thing as a green star. Red or orange or whitish or bluish, but not green, because that curve is determined by the laws of physics that say when you heat something up, the radiation that's given off by a body is determined by a certain set of rules. It's all part of quantum theory. We're not going to go into the details, you'll be glad to know. But it means that a star can't simply decide, I want to glow in the green part of the spectrum. That's not allowed by the laws of physics. So if you heat something up, what happens? Well, if you heat it up, it goes a dull red, and then you get it hotter, it goes orange, and you get it hotter, it goes sort of yellowish, and you get it hotter, it goes whitish. It's the same sort of idea. No matter how much you heat up an iron bar, it doesn't glow green, regardless of what temperature you heat it up to. You go through a certain sequence of colors, but not the sequence that you might think. It doesn't go from red to green to blue. It goes from red to blue via white, not via green. Let's have a look at why that is. Again, we're not going to go through the details, but this is a rather neat little educational diagram provided by PHET. You can go and look that up again if you wish. This is looking at how much light is given out on the vertical axis as a function of wavelength. 0 means the ultraviolet end of the spectrum. The visible part is clearly where the rainbow is, and everything to the right of the rainbow is infrared wavelength. The 3 simply refers to the wavelength in microns. So we've got a fraction of a micron in the visible part of the spectrum and 3 microns on the right-hand side. And this is showing us how much light is given out at each wavelength as a function of the temperature of the object. So this is a so-called black body spectrum. Black body meaning it's not an object that has any color, because that would distort the readings we're getting. It's a black object which is heated up until it glows because it's hot, and we're looking at how much light is emitted by this black body object. So if an object is at 1200 Kelvin, so that we would already think of that as quite hot, but of course it's very low heat as far as a very low temperature as far as a star is concerned, then most of the radiation is given out in the infrared part of the spectrum. What's going to happen is I'm going to take the temperature up and see what happens to this orange line. Two things are going to happen. The orange line gets higher because more radiation is given out when something gets hotter. And the peak, where is the peak part of that orange line? That's going to shift to the left. And that's because when objects are relatively cool, they emit infrared light, but as they get hotter and hotter and hotter, they start to emit more visible light, more ultraviolet light. So the peak starts to shift to the left. The fundamentals of why are buried deep in the heart of quantum theory, which we're not going to go into, but we'll see the results here. So let's go to a higher temperature. So the orange line is now higher, and it's moved slightly to the left. And we're now getting a little bit of radiation at the red end of the spectrum, notice. Most of it is in the infrared, but there's a little bit of radiation at the red end of the spectrum. So when we heat something up to this temperature, it will start to glow red. What's going to happen if we go a little bit higher? I'm going to try and avoid, although this orange line is going to keep going higher, I'm going to keep rescaling this so it doesn't go through the roof. Otherwise, it's difficult to see what's going on. I'm going to take the temperature up. It's now 2000 Kelvin. So where are we now? We are still pushing out most of our radiation in the infrared part of the spectrum. But notice we're now getting substantially more at the red end of our visible spectrum. Essentially nothing in the blue, but quite a lot in the red. We're now at 2000 Kelvin. If we bump it up to 3000 Kelvin, we're at the sort of Betelgeuse level now. We're now at the red giant sort of stage. The peak is still just about in the infrared. The peak of a red giant's output is very close to the red end of the spectrum. So a huge amount of light is coming out red, a little bit of blue, but it's completely dominated by the red part of the spectrum. So red giants, not surprisingly, are red. If we take it up one stage further, what happens if we go to something like the temperature of the sun? Now we've pushed it so that the peak is now in the visible part of the spectrum. How much light are we getting at all wavelengths? We're getting shed loads of light at all wavelengths. We're getting a lot of red, a lot of blue, a lot of green, a lot of yellow. We're getting essentially white light. So you might say the sun looks just a little bit yellowish, but actually the sun is simply emitting at all visible wavelengths. Our eyes are more sensitive to some wavelengths than others, but the sun is emitting at all wavelengths because this peak effectively gives you a large value throughout the visible part of the spectrum. What happens if we take the temperature up higher? If we go to 11,000 Kelvin, this is a sort of an A-type star, if you like, then you can see the peak is now pushed into the ultraviolet part of the spectrum. So what does that mean? Well, it means we get much more light in the blue end of the spectrum than we do in the red end of the spectrum. So yes, the star is emitting across all wavelengths, but much more blue than red. So this star is going to be white, but with a tinge of blue because there's much more blue than there is red. So that's why we talk about A-type stars as being bluish white. There's a lot of light across all wavelengths, but there's more blue than red. And if I just go backwards and forwards, you can see that when we're dealing with red giant stars, it's more red than blue. When we're dealing with something like the sun, it's essentially all wavelengths. When we're dealing with an A-type star, much hotter star, more blue than red. So it's either reddish or white or bluish. Never is it green but no red and blue. We can't ever get a curve which peaks in the green and dies in the red and the blue end of the spectrum. That peak is never that sharp. It's never that spiky. So we will always get one of these types of results, mainly blue, white, or mainly red. And that's why we have no green stars. That's why when I showed you the stars in the color space, it only follows that curve. It doesn't just arbitrarily go all over the color space. And if we were to map them out, we see that, of course, perhaps you're familiar with the Hertzsprung-Russell diagram, we can map out the stars and we see some of them are red giants, some of them are red dwarfs, some of them are white dwarfs, some of them are bluish at this end of the spectrum. If we think about the colors down at this bottom end, it's very similar to what I showed before in terms of heating up an object. It goes red, then orange, and then sort of towards white. So that's pretty much the same. We don't tend to have objects on Earth which we heat up to 10,000 Kelvin. They don't tend to survive heating up that much. They tend to vaporize. But we can see that lots of stars can be mapped out, and we get star colors all the way from mainly red to whitish to mainly blue. But a little while ago, somebody asked themselves, well, there are stars all over the galaxy, and indeed in many other galaxies, across the entire spectrum here. In other words, surface temperatures varying from a few thousand to tens of thousands of Kelvin, and hence surface colors that seems to be reddish or whitish or bluish. But what's the average? What is the color of the universe? If you looked at every star in the Milky Way and every star in millions of galaxies, what color overall are they? Some people with far too much time on their hands decided to actually calculate that. They tried summing up, mathematically speaking. They didn't do this photographically. They looked at the spectra of lots of stars in lots of galaxies and basically said, well, if that's their spectrum, then this is how the light is distributed at different wavelengths. Let's add this up for millions and millions and millions of stars to get the average color of the universe. And depending on this projector, we will see what color it actually produced for the final result of adding up light from 200,000 galaxies within 2 billion light years. So I don't know how many stars contributed to this color, but probably a few billion in each of those 200,000 galaxies. So there's probably 1,000 trillion stars adding up to give you this final color. And the final color was beige. Off-white, if you prefer. They didn't want to publish a paper saying the universe is beige, so they came up with different names. They called it either cosmic latte or, my favorite, skyvery. I like that one, skyvery. So yeah, it's a little off-white, and perhaps that's not surprising that we've got lots of red stars, lots of blue stars, but the numbers are not equal. There's an awful lot of red dwarfs. There's a huge number of red dwarfs in our Milky Way and probably other galaxies, but they are fainter than the blue stars, which tend to be hotter and more luminous, and it looks like they sort of balance out. Less blue stars, but they're really bright. A lot of red stars, but they're quite faint. When you add them all up, it's sort of neither one nor the other. It's somewhere very boring beige. So that's how stars work. What about nebulae? Nebulae are colorful, but for different reasons. Let's see if we can understand the difference. If we're looking at stars, or indeed galaxies, and for this purpose, I'm going to consider galaxies as simply collections of stars. They're not. Galaxies are collections of stars and dust and gas and other things, but let's just assume that most of the light of galaxies is coming from their constituent stars, and we've seen how stars behave. So galaxies are sort of the same thing. They have a continuous spectrum. If we were to look at how much light is coming out as a function of color, we get colors across the whole spectrum. You could argue that on average it's sort of skyvery, cosmic latte. So yes, individual stars might have more red than blue. Individual stars might have more blue than red. But generally speaking, there are colors across the whole spectrum when we look at a galaxy. But if we look at a nebula, and it's easier to see nebula in our own galaxy, but this would be true of any nebula in other galaxies, when we're looking at a cloud of gas, we find that only certain wavelengths seem to appear in the spectrum. If we take a cloud of gas that's being heated, perhaps by a nearby star, we will see particular lines. This is supposed to be just representative. Don't worry about the actual colors. This one just happens to be a spectrum in which there's one clear red line and a couple of blue lines and a violet line, etc. If that nebula was actually blocking light from a more distant source, then it actually might show up as an absorption spectrum rather than an emission spectrum. So this is simply a cloud of gas that has been heated up, and it's not radiating at all wavelengths, it's radiating at very specific wavelengths. So can we understand that difference? Again, we're not going to go into all of the details of quantum theory, but it's based on the principle that nebulae are clouds of gas, and gas is made of atoms. Now, we sometimes think of atoms as being, well, a positively charged nucleus and a load of electrons buzzing around it like bees in a beehive. Well, that's not really a fantastic way of thinking about it. It's very much a sort of a kindergarten view of atoms. It's the picture we tend to have in our heads, but it's not useful for a number of reasons, not least because all the electrons look the same here. In other words, it would appear that all the electrons have the same energy because they seem to be all doing the same thing at the same distance. It would actually be more useful to think of the electrons in atoms as, well, it's not quite a solar system, but it's perhaps more like a solar system in the sense that some electrons are close to the nucleus and some electrons are further away from the nucleus. It's still not right, it's still a cartoon, it's still not exactly the way atoms work, but it's more useful because at least it does remind us different electrons have different distances to the nucleus, and therefore they have different energies. Now, in an atom is not a flat pancake like a solar system, so we really should be thinking about shells of electrons. But again, shells are more difficult to draw, and it's a lot easier to draw an atom thinking of it as if it was a two-dimensional object that we can sort of image from above. It's just a convenience. It's not reality, it's just a convenience. But at least it does tell us that we have different energies. And in physics, different energy for an electron moving in an atom, the amount of energy that it has, the amount of energy that it might give up to a photon of light leaving or arriving at that atom, the amount of energy is related to the color. Again, I'm not going to go into the details, but photons, light of the red end of the spectrum has a lower energy than light at the blue end, the higher energy. And so in other words, a 400 nanometer quantum of light, a photon, a particle of light at the blue end of the spectrum, 400 nanometers, has a higher energy than at the other end. We're not going to worry too much about the numbers. What matters is that every atom has a particular collection of electrons at a particular set of energy levels. So in other words, we can sort of draw a picture like that and say that represents an atom, in this case quite a complicated atom because there's an awful lot of electrons in there. And the way these electrons are arranged in the atom are specific to the atom. That's determined by quantum theory. That's one thing quantum theory tells us, is how atoms are put together. So one atom might have a particular arrangement of electrons, and a different atom would have a different arrangement of electrons in the atom. And hence, we get a different fingerprint of where those lines form. Because remember, different arrangement of atoms means different energies. Different energies correspond to different colors, as I've shown in the last diagram. So each atom has its own fingerprint of where we would expect to see those lines. If the gas comprising these atoms is heated up, we might see emission at particular values. If the gas is sitting in front of a star that's trying to shine through the cloud of gas, it might absorb particular wavelengths, and hence we see an absorption spectrum. But the point is, the pattern of where these occur will depend on the atoms that are in the gas cloud. So why the difference? Just to summarize, light from a nebula is made of discrete colors because that corresponds to the discrete energy levels of an atom. Whereas light from a star is continuous. All colors are present, even if in some cases there's more red than blue or more blue than red, all colors are present. But why the difference? Because they're all made of the same stuff. Nebulae might be made of hydrogen, helium, oxygen. Stars are made of hydrogen, helium, oxygen, etc. So why the difference? If they're made of the same stuff, why do clouds of gas behave differently to stars when it comes to looking at their colors? The fundamental difference is how many atoms we're dealing with, the density. If atoms are well separated from each other, they don't really know the other atoms exist. If one atom is here and the other atom is a micron away, on a quantum mechanical scale, a micron is a huge distance and one atom doesn't know about its neighbors. And each atom emits a spectrum that's determined by the rules of quantum theory. But if you put the atoms close together, like in the middle of a star, for instance, they are continually bumping into each other. And so, in effect, the spectrum of an individual atom gets smeared out to the point where the entire spectrum gets populated with emission at different wavelengths. So what matters, if we want to understand why some things seem to give continuous spectra and some things give discrete spectra, we need to have a think about, well, how many atoms are we dealing with? Let's just think about how many atoms there are per cubic centimeter of various objects. If we take a lump of metal, or indeed almost anything, we could take a lump of glass, for instance, a lump of metal, even a lump of wood. How many atoms are there in a cubic centimeter? It's a big number. It makes a slight difference which metal you choose, whether you're picking aluminium or lead, but it doesn't make that much difference. It only makes a tiny difference to the last zero. Generally speaking, we're talking about a huge number of 100,000 million atoms per cubic centimeter. What about the Earth's atmosphere? There's clearly less atoms per cubic centimeter in the air through which we move so readily. Still a big number, still a huge number, but it's obviously less than the number in a lump of metal. But it's still large in the sense that the molecules in the air are continually banging into each other. What if we take the surface of the sun, the photosphere, the visible surface of the sun? Perhaps it's surprising to find that there's substantially less material there than there is in the Earth's atmosphere. It's a lot hotter, but in terms of the number density, how many atoms are there? Substantially less on the surface of the sun compared to what's going on in this room. If we were to go further out into the sun's corona, then it's definitely starting to fall off quite dramatically. If we go out into the solar corona, which we see with the solar eclipse, for instance, now we've gone down to a very modest 10 million atoms per cubic centimeter. If we go to a typical nebula, there's a lot of variation between the heart of a very dense nebula and a very diffuse nebula, but just typically we might be talking about 10,000 atoms per cubic centimeter, substantially lower still. And if we take the interstellar medium, if we just arbitrarily go, let's say, halfway between stars, just pick a couple of stars at random and look at halfway between them, the interstellar medium might have 1 atom of something per cubic centimeter. And the ultimate is, what if we take the intergalactic medium? What about if we go halfway between here and the Andromeda galaxy? Well, I've written it as 0. Nothing is ever 0. What I mean is you're unlikely to find 1 atom in any given cubic centimeter. The actual density in the intergalactic medium would be something like 1 atom per cubic meter. So in other words, this number ought to be more like 10 to the minus 6, 1 part in a million. So there's a huge difference between how many atoms we're talking about in a given volume on going from, let's forget the top two, the sun's atmosphere, the sun's surface. There's a huge difference between that and thinking about a nebula in which there are very few atoms in a cubic centimeter. And that is why the more dense ones have a continuous spectrum. That's why the surface of the sun has a continuous spectrum. Whereas if we go to the solar corona or a nebula, then we get such low densities, the atoms hardly ever bump into each other, which means each atom is behaving, as it were, independently of all the other atoms, and they just give a spectrum which is dictated by the laws of quantum theory, which says that they just radiate at specific energies, specific colors, corresponding to which atoms we're dealing with. And we can tell by looking at the pattern, is this hydrogen? Is this helium? Is it oxygen? Is it nitrogen? That's how we tell what these clouds of gas are made of, simply by looking at the pattern of lines that we see in these emission spectra. So we've had a good think about color in the cosmos. We've thought about the colors of stars. We've thought about the color of nebulae. Let's just finish with some thoughts about color. You get this question so many times when you're talking about color images. It's sometimes from amateurs, but in particular when you're looking at images in magazines or on web pages. Are the Hubble Space Telescope images that have these incredible views of various objects, are they true color? Well, some of them might be, but most of them aren't, especially when you're looking at nebula. If you're looking at a galaxy, it might well be that that image of the galaxy is the true color of the galaxy, the actual color that you would see if your photoreceptors in your eyes were sensitive enough. But when it comes to imaging nebulae, what the scientists want to do is try and tease out as much information as possible from the image. So a nebula like this, the Pillars of Creation in the Eagle Nebula, M16, they think about which particular wavelengths of light. Hubble, of course, is restricted not strictly to visible. It can look a little bit into the ultraviolet and a little bit into the infrared, but it spends most of its time looking at the visible part of the spectrum. The constituents of this particular nebulae are mainly sulfur, hydrogen, and oxygen. And that shows you the color at which sulfur glows, the color at which hydrogen glows, and the color at which oxygen glows. In other words, 3 very specific wavelengths. If we could isolate them, if we could only take a gas cloud of sulfur, a gas cloud of hydrogen, a gas cloud of oxygen, we would see lines at those particular points. Possibly elsewhere as well, but in particular, those 3 wavelengths. Notice that sulfur and hydrogen glow with a very similar tint of red. So if you were to simply say, we're going to take a color image of the Pillars of Creation, you would not really be able to tell the difference between, is that stuff over there hydrogen or is that stuff over there sulfur? Because it would look pretty much the same. It would be almost the same shade of red. Oxygen would be fairly obvious because that's a sort of a cyany color, but sulfur and hydrogen would look the same. So when it comes to making a color image, they take an image using a sulfur filter, letting through only that light. They take an image with a hydrogen filter, letting through only that wavelength. They take an image with an oxygen filter, letting through only that wavelength. And then they say, right, we're now going to make a color image. And the sulfur is going to be colored red, the hydrogen is going to be colored green, and the oxygen is going to be colored blue. Why do they choose that? They try and keep them in the right order. In other words, in terms of wavelength, that's the longest wavelength there, that's the shortest wavelength, and that's red, green, and blue. So they keep them in the same order as the original wavelengths of the filters they used. But clearly, hydrogen isn't green. But sulfur has already used the red filter, sorry, the red channel of the color, so it can't use red, so they put the hydrogen in the green, and oxygen gets its blue, which is relatively natural anyway. And if you do that, you end up with a color image that looks like that. The pillars of creation are not that color. If you've ever tried photographing them yourself, you find that they come out red. They come out red because there's sulfur and hydrogen, and there's a little bit of oxygen in there, but most of the light is red because it's basically a hydrogen nebula with other stuff in there. But by assigning this to red, this to green, and this to blue, we're able to see subtle variations in color because our eyes are very good at seeing variations in color because we've got neurons in the back of our eyes helping us do that. And so we can start to see, the subtle variations in color in there, which I can then figure out, or scientists can then use to say, I can now see that there's hydrogen here and sulfur there, and I can see variations. So it's not true color, but it is scientifically useful to be able to do that. And the converse of, if you're not allowed to do that, but you can only take in true color, you lose the science, you lose the detail, you lose the contrast, because everything looks red. The entire scene there would look essentially red, and it would be much more difficult to pick out the detail. So no, this so-called assignation of sulfur and hydrogen and oxygen to red and green and blue is a particular way of processing images for nebulae. It's called the Hubble palette. It's so common, they've given it a name and they've called it the Hubble palette, and some amateurs prefer to do the same. Some amateurs will take their images and they will buy filters that let through sulfur and hydrogen and oxygen. They will take those three images, they'll put them in Photoshop and assign them the red, green, blue channels, and out will come an image that looks a little bit like a Hubble image. So people, as well as amateurs as well as scientists on the Hubble Space Telescope, will do precisely that in order to pull out as much detail as possible. And of course, the electromagnetic spectrum is not limited to the visible part, which we know and love, because although the Hubble does do a little bit in the ultraviolet, it doesn't tend to go in there much. And of course, we're now used to the idea that Herschel and Spitzer and James Webb have been operating in the infrared. And so, of course, the James Webb Space Telescope, none of its images are true color, because by definition they can't be. It's spending all of its time working at various wavelengths in the infrared part of the spectrum, but it is doing exactly the same as the Hubble. It takes an image at this wavelength, that wavelength, that wavelength, and trying to keep them in the same order, it assigns them, this is going to be red, this is going to be green, this is going to be blue, and then you get a color image which hopefully looks nice. And our eyes are then able to see those three colors and distinguish what's going on at those three wavelengths. This is the filter wheel, one of the filter wheels of the James Webb, very complex, lots of different filters that operate either across a broad range of colors, as indicated on the bottom left-hand side. Some of them work on very narrow ranges. In other words, they only pick out infrared wavelengths that are known to be interesting for some scientific reason. So some of these filters are very narrow, some of them are medium width, some of them are very wide, and depending on what science you want to do, the person taking the observations will choose whichever filter they want to be in front of the camera or imaging system that they're going to be using to decide what science they want to get out of those particular images. So let me summarize. The eye is not a camera. It is far more sophisticated in many respects, simply because the eye is doing a lot of processing. It does not simply detect intensity of light and send those signals to the brain and tell the brain, there's the signals, get on with it, figure out what the hell I'm looking at. No, the eye does a lot of the heavy lifting by filtering out what it thinks the brain needs. A lot of that information is filtered by the eyes. They do some processing, and only a tiny fraction of the actual information hitting the retina actually gets sent to the brain, so that the brain has less work to do. That's a result of evolution of the way our eyes work. The colors of stars are determined by so-called black body radiation, and star colors will range from red as we go up in temperature, from red to orange to white to bluish white, never going through green. Hence, whenever we look at images of the sky, we tend to see blue stars and red stars and sometimes white stars, but we never see anything that is emerald green. The colors of nebulae are determined entirely by the rules of quantum theory, quantum jumps between electrons in the atom. Every atom has a different set of energy levels, and hence the light that is emitted or absorbed by those atoms will be a certain set of energies, and that corresponds to a certain set of wavelengths, which corresponds to a certain set of colors. So given atoms will radiate or absorb at given colors, which is really useful because if we take the spectrum of a nebula, that means by looking at the pattern, we can work out what the nebula is made of. I'm not going to talk in any detail about spectroscopy, but we know that if we take a spectrum, we can split it into its colors. It's not only useful for telling what things are made of, we can also tell how fast things are moving, because if one star is going around another, or it's wobbling because there's an exoplanet moving around the star's moving towards or away from us, we get a Doppler shift, a slight variation in the position of these absorption lines, which allows us to get the speed of planets or stars, or if we're looking at very remote galaxies, we get effectively the speed of recession because the universe is expanding. So spectroscopy and splitting light into colors is extremely useful on a cosmic scale as well as a planetary scale. But the take-home messages are the eye is not a camera, and nebulae and stars behave differently because we're talking about different densities of objects. Thank you all very much.